Welcome. My name is Lynne Lowry, and I'm the Senior Vice President of Business Development for StarFish Medical. StarFish Medical is Canada's leading medical device service provider, offering a full service complement of design, development, and manufacturing services in Victoria and Toronto. Our goal is to help clients create world-class medical devices. Over the past 5 years or so, StarFish Medical has held an annual Playbook events in Toronto or Victoria that provided a forum for entrepreneurs, CEOs, and industry leaders to share their success stories, challenges, and lessons learned through presentations, panel discussions, and various networking events. Due to the COVID pandemic, most in-person events canceled this year, including our events. We decided to try something new as an alternative to support our current Playbook community and expand our network and reach by launching a virtual Playbook event. Our first virtual Playbook event was held in late July and featured a strong lineup of speakers discussing how COVID is changing medical device commercialization. It is with pleasure that I welcome you to StarFish Medical's second virtual Playbook webinar series on new opportunities and challenges in microfluidics. You will notice a few polls pop up between speakers, hopefully only when I'm talking, and we would really appreciate if you could answer the polls. That allows the speakers to see the makeup of our audience today, since we can't see everyone in the room. It is sort of our virtual way of making eye contact with you. We have a fantastic lineup of speakers today that I promise will stimulate new ways of thinking about microfluidics, your business model, and likely generate a thought-provoking Q&A session in the wrap-up. Our first speaker is Stevan Jovanovic, who is the CEO of S2 Genomics. Previously, Stevan was the president, CEO, founder, and CTO of Integenics, which is the global leader in rapid human identification and now part of Thermo Fisher. Dr. Jovanovic is the author of 28 peer-reviewed papers and an inventor of over 140 international patents, in addition to being considered a technical expert in the fields of genomics, rapid DNA, microfluidics, and system integration. Dr. Jovanovic will be speaking about microfluidics in the real world. Welcome, Stevan.
Thank you, Lynn, for the introduction. So, and by the real world, I mean getting products to people to where they can use microfluidics to change things. So microfluidics, just as a first speaker, quick introduction, volumes less than typically a microliter, and in dimensions less than a millimeter. And so you have small volumes, which can lead to reagent reduction. It can be the right size for small samples like biopsies. It's used frequently in injecting samples into an analytical platform like a mass spec, and it can also help you miniaturize cartridges. One thing that you're going to see throughout probably all the talks is surface chemistry will either be your friend or your foe. The challenges are either you need a sample to answer system, and I'll show you one example of that, or you need to fit in both the upstream and the downstream workflows. So that's one challenge. The other is going from the macro scale where your sample begins into the micro scale. How do you interface? How do you mix? Bubbles are always an issue. And system integration is key. You'll need know-how, tool chest of components, hopefully some IP, and then how to fabricate prototypes and scale up to manufacturing. And we'll talk more about that later. So the first thing is, do you need microfluidics? If you need it, what processes must be in the microfluidic realm? And are those going to be passive or active? Where active are on device pumps, valves, sensors, things of that nature. And then what processes can be done in the instrument and what need to be done on the microfluidics, and then how do those work together? So I'm going to set the Wayback Machine for when I entered microfluidics. I joined a company called Molecular Dynamics, which was creating the next-gen sequencer at the time, which had 96 capillaries, and those had a 75 micron ID. So that's a case where the surface chemistry was a challenge and needed to be solved. but that's another story. What I joined Molecular Dynamics was to bring in sample prep for the megabase, and we wanted to reduce the amount of reagents used so that we could reduce the cost per sample. By the way, the megabase, about a third of the human genome was done on this platform. So what I came up with, and which was funded by NIH, was an approach for a flow-through microfluidic system. So where we do nanoscale reactions inside capillaries in boluses. And it's passive because we're going to use syringe pumps to move the fluids around. There's 3 sample prep repeat units that are shown here. There's a PCR module, a cleanup, a cycle sequencing, and then another setup to inject into the array of capillaries. And the beginning part of this was, once we came up with the idea, we had to come up with the components. And in this case, it was inventing the components. One of those I'll point out is what I call the Frolic fiber optic-like capillary connector. So this is an FC connector that you can see on the left side when we split it open like a book. That you have black is plugged and open is the circle, and they can be twisted so they can either be in configurations that they line up as shown on the bottom or not at the top. So you can have a closed or open valve, or you can route things with this. We extended these to array connectors, and you can see here's one version, and then we took a commercial fiber optic one and put in capillaries. These work very well. And then we also built an injector, so to inject the sample into the CE. This is a 100 nanoliter injector, and a little bit of data down below. So that was all nice. And then we built systems and tested, and which is where I recommend you start. You have to show that your parts are going to work. Here you can see 2 syringes on syringe pumps with these Frolic connectors going into a device where we're mixing things, going into a thermocycler. And we ran this and got pretty good results, but there were some challenges with this. And the main one was that there are a lot of parts in it. There's also concern about controlling those boluses and crosstalk. And then the other part is, in one solution, we would hardwire this into a megabase, which would make a very complex machine. So I was working on the grant renewal and thinking hard, and came up with the aha moment. And we basically changed courses. And this shows a capillary cassette that we call NanoPrep. And this is a passive microfluidic device also. This is a 2x8 array of capillaries, or 500 nanoliter capillaries. There's a board here so that you can spin things out by flipping this onto a microtiter plate and actually spin the liquid out. And it's spring loaded, so it will seal nicely in thermocycling. So this works very simply, and when you can get the simple and elegant, it's well worth the thought process. And in this case, we would take the DNA, and this is to do a cycle sequencing reaction. So you would put in a PCR product, for example, with a chaeotroph, dip your capillary in, it fills by capillary action, very simple. The DNA then prefers to be on the glass surface rather than in the liquid, so it binds to it. So there, that surface chemistry is working for you and will turn out to be a big advantage. We then would clean it out and leave the DNA on it and dry it, and then dip into sequencing reagents. Again, it would fill by capillary action, and then it would be ready to go. So this solved both the macro-micro interface, and it solved the mixing, because the DNA is on the inside, the reagents can get to it, and that surface chemistry will normalize the template concentrations. And you'll see the advantage of that in a moment. So this went through a cycle. So the 16 channel was our beginning point. We built a air-based thermocycler and a wash station, went from 16 to 96 to 384, and this actually went into product. These are actual pictures of the product, which is another story. So here's the performance advantage. This is showing in the megabase, and the pink line is the performance of the megabase over a range of concentrations, and you can see it's got a sweet spot that's sort of narrow. When we go to the template capture using NanoPrep, it brings the bottom up from no read length to a pretty good read length, and then it flattens out the curve, and this extends out to the right for at least another 20-fold. So they're basically, you can no longer overload the system at all. And not only did that improve the range, it improved the success rate. So samples that were difficult worked. So that's an example of passive microfluidics. Active microfluidics, I started a company called Microchip Biotechnologies, which turned into Inogenics, and this is our first product that we produced there. Again, it's for cycle sequencing for Sanger, and in this case, we automated both the cycle sequencing and bead-based cleanup. You can see this is in a robot, so here's a station where we're cleaning out the pipetting head in between sessions of using it, and then here's the glass microchip. So this has little move valves. So these are pneumatically actuated valves. You can see the pneumatic interfaces on the right. So any 3 valves will make a pump. So not only could we close things and open them, we could use them as pumps. We'd add the reagents with the robot. So that's our macro-micro interface. And we did both spray cleanup and thermocycling here. And if you can play the video, first video here, show about 30 seconds of this in operation. This is actually the second one. We can go back to the first one. There we go. Okay, so this is shows a zoom in, and what you're going to see here is that's an individual valve. The samples coming from the upper right, the reagents from the left, and you can see that when they go together, they don't particularly mix as they go through the first valve. But when you come into the second valve, you have the 2 streams that are pretty much one on top of the other, and this goes from a pancake to a hemisphere to a pancake. And when it does that, you get nonlinear forces on it, and you can see by the time you get to the next valve, things are pretty much mixed, and by the time you get to the reaction chambers, they work perfectly. So if we can go back to the presentation, you know, if I control that. Okay. So in this case, we use these move valves as valves pumps and for mixing, and also started to use paramagnetic beads, which are one of the key tools that I recommend you look at if you're not already using them. We also use the move valves in, I think, what's probably the most complex sample to answer system that's been fully integrated. This is the rapid hit. And we basically took a whole forensics lab that you can see on the top for doing reference samples, and we put all the sample prep onto this one cartridge, and the instrument has an 8-channel capillary array electrophoresis. So the way this works is there's about 120 or so valves on this, so we can both pump and close things off. The user would put a sample such as a swab or another type of sample in this lysis chamber. This is a reagent pack that the instrument would open up, and basically we would move lysis buffer using these move valves up to the lysis chamber. We would heat that up to about 70 degrees to free the DNA from the cells in there, and then we would use the move valves to pump them down to a chamber that was preloaded with paramagnetic beads, and these are spry-like beads. The DNA would be captured onto that because it would be in a chaeotroph, which was in our lysis solution. And again, the DNA would prefer to be on the bead than in the chaeotroph. And then we could do 2 sets of washes with that. Following the wash, we would move it down to an STR reaction chamber. And again, we would capture the beads using a magnet down there. We could then pump in STR fluid, and this was loaded onto the cartridge in miniaturized valves that we'd created. We then sealed off the reaction chamber because we're going to do PCR for these STRs. Is basically a 16-plex or 24-plex PCR. And after that, we'd open the valves, move it to our size standard was, mix that in, and then inject it into the instrument where there was the capillary array electrophoresis. So that's now a Thermo Fisher product. And then now I'll switch to the last subject, which is single-cell genomics, which is what we're doing now. And the real breakthrough here was a microfluidic device. So this is shown from the Microscope paper. There's 2 papers came out the same month back-to-back in Cell. And basically, I have lost the screen. Let's see, there we go. You would have cells come in one channel, beads in the other, and you'd want to get it, they would then be pinched off into a miscible fluid, and you'd get a droplets, these nano droplets that would hopefully have one and only one cell and one bead. After you do the first reaction in there, typically binding cDNA onto the bead, which has barcodes, you'd break the beads, mix everything together, do your library prep sequence, and then you'd decode by using the barcodes. And this is 10x technology, and Dolomite both have products in this space. So what my current company is doing is we're making those single cells and nuclei. We have a device called the Singulator that automates that whole process. And if we can go to the next video, this will show you a little snippet of it. And this is, most of this happens in the macro scale, but there's also micro scale features in this. So this is an injection molded cartridge that we're putting in a whole mouse kidney. This is a camera inside the instrument. We're going to make nuclei. In this case, we add a nuclei isolation solution. This has a rotor that you can see spinning. There's micro features on the bottom to disrupt the tissue. And then on the sides of that white spinning piece is a gap. That's a 150 micron gap. And you can see the material ends up on top of that. It's then pulled into the right side, washed, and you pipette it out, do a central, one quick centrifugation, count it. And this is what you want for nuclei. You want them to be dead. We get about 35 million out of a single mouse kidney. And you use about 100,000 for a reaction. So if we go back to the last couple of slides here. So, so this works with all the analysis platforms for single cell and also for FACS. We've used it to create TILs. If anyone in the audience is interested, please contact me. It's a great opportunity. So to wrap up, microfluidics can give you compelling advantages, but it also comes with challenges. I'd urge you to carefully look at your workflow, divvy up what goes on the cartridge, what goes on the instrument, and then you have your own learning curve and challenges. So this is where working with a group like StarFish will be very beneficial to you. Thank you.
And I just wanted to let everybody know, if you have any questions for any of the speakers, if you will just put them in the chat, we hope to get to these by the on the Q&A at the end. So thank you very much. Our next speaker is Dr. Shana Kelly, who is a professor at the University of Toronto and a member of the Department of Chemistry, Biochemistry, Pharmaceutical Sciences, and Biomedical Engineering. Her work has been recognized with a variety of distinctions, including being named one of Canada's top 40 under 40 and a top 100 innovator by MIT's Technology Review. Additionally, Dr. Kelly is a founder of 3 molecular diagnostic companies: Genome Sciences, acquired by BD in 2005, Exogenic, acquired by General Atomics in 2017, and Cellular Analytic. She is an inventor with over 50 patents issued worldwide. Dr. Kelly will be speaking today about visualizing rare cell biology. Welcome, Dr. Kelly.
Okay, thank you, Lynn, for the introduction, and thanks so much to StarFish for including me in this event. What I'd like to do today is to tell you a pretty quick story about a new microfluidic technology that's been developed in our lab and how it's being applied to elucidate rare cell biology and also apply insights from single cell measurements to drug development and disease screening. So before I jump into the technical part of the presentation, I'll just give you a really brief overview of my group and some of the things that we work on. My lab at the University of Toronto is quite multidisciplinary. We have chemists, biologists, engineers, all working together on important problems. We really focus on platform development and technology development to provide new solutions for diagnostics and drug discovery. And we focus not only on producing high quality academic science, but also on turning our research breakthroughs into commercial products and systems for healthcare. And one of the core areas of our expertise is the development of electrochemical sensors for point-of-care diagnostics. Over the years, I've been involved with the creation of 3 companies in this particular area. Genome Sciences, that was acquired by Becton Dickinson in the mid-2000s. Exogenic was spun out of our labs in 2010 and acquired a couple of years ago by the US defense contractor General Atomics. And then very recently, we've started a new company, Arma Biosciences, that is commercializing a handheld device for direct detection of the SARS-CoV-2 virus directly in the oral cavity, kind of enabling real-time measurements of a viral infection without the need to draw a sample. But today I'm going to focus kind of on the other side of our group, which is on the development of new microfluidic technologies. And this particular one was developed to allow us to locate and profile rare cells in complex samples. This technology is being commercialized by a company that was founded last year. It's called Cellular Analytics. And in this short presentation, I'll tell you a bit about where the company is taking the platform. As we all know, disease biology is extraordinarily complex. The response of the immune system to infectious disease or the presence of a cancerous tumor can vary significantly among individuals, and there's tremendous heterogeneity at the cellular level that dictates how disease progresses. As we continue to advance our understanding of disease at the molecular and cellular level, it's increasingly important that we have tools that allow us to seek out rare cells that could be driving the progression of disease or dictating how an individual may respond to treatment. Measuring the genotypic and phenotypic properties of rare cells that are present in tissues or different types of living systems presents a significant technical and technological challenge. While our community is making tremendous progress as a whole towards the development of systems for single cell measurements, you just heard about some very powerful technology for single cell analysis. Most of the technologies that we have access to are not able to provide accurate information regarding molecular level changes in rare cells in the presence of an overwhelming excess of normal cells. And this is the situation that really describes most samples and systems where we can do experiments that help us elucidate the foundation of disease biology. So developing a platform that could not only isolate, but also functionally profile rare cells was the goal of the work that I'll talk you through today. Our approach, which we trademarked as Cytofind as we've advanced it towards commercialization, works in a manner that's somewhat similar to flow cytometry. So in a flow cytometry experiment, you use a fluorescently labeled antibody, you label cells, quantitate protein expression. But this type of approach has limited utility if you have a very low cell count or if you have a large excess of a non-target cell. So what we do instead is to use very small magnetic nanoparticles that stoichiometrically label cells and will allow us to read out levels of protein expression. The challenge of using these small nanoparticles is that they're difficult to use for cell capture. And so we want to use the nanoparticles to kind of bring the rare cells out of the background of non-target cells, but then also be able to quantitate levels of protein expression. So this is really where microfluidics comes in. So after we incubate the cells with magnetic particles that are labeled with an antibody that targets whatever protein we want to profile, we flow them through the device shown as kind of that large rectangle at the top of the slide. And this device contains these X-shaped structures, which you can see over on the right-hand side of the slide. And it's these X's that allow us to capture the labeled cells, even though we don't have a lot of magnetic force to work with, given our small magnetic nanoparticles. And so we use these X's to locally slow down the flow of the solution while we keep the overall flow and sample throughput high. So as the solution is flowing through, it's getting slowed down every time it encounters an X. And in those regions of low flow, we're able to capture our labeled cells. And then in order to read out protein levels, we create an array of different capture zones within the device. This can be done by patterning micromagnets around the X-shaped structures and varying the size of the internal magnets to create a capture gradient. Or we can introduce small variations in the depth of the channel that alters the solution velocity within the device very incrementally and creating many, many different capture zones. So we can use either of these approaches, create devices that have 100 or more capture zones within them. And then as we flow the sample through, cells that have a high level of labeling will get trapped in the zones closest to the inlet. Cells with a low level of labeling will travel through the device and get trapped in the zones at the end of the device near the outlet. And so to visualize the cells after we process the sample, we immunostain them. So this allows us to distinguish target from non-target cells, and we use a cocktail of stains to accomplish this. Once we visualize the cells on the device, we can generate a histogram of single cell level data. So that's what's shown in that bottom graph. The overall position of the histogram, so we're just picking off how many cells, usually single digit numbers of cells that we see in the different capture zones of the device. The overall position of the histogram gives us an expression index for the population of rare cells, and it also gives us a sense of the level of heterogeneity overall in the cells that we're analyzing. So we could pull up the first video. I'll just give you a sense of how this works. I think no microfluidic talk is complete without a few videos. In this first one, it's actually the other one. What you're going to see is whole blood flowing through a device and flowing past one of these X-shaped structures. So this is the right video. And so you can see the complexity of the problem here. So there's billions and billions of blood cells, or we're seeing billions of blood cells that are running through the device. And then in this particular experiment, we're trying to capture small collections of cancer cells, and we're trying to pull them out of the vast background of the blood cells. So just go to the next video. We can just cycle through this one to the other one. And so after we process the sample, you can see that we're left with this small collection of cells captured in a low zone device around the X. And then we do our immunostaining and then generate that histogram. We can just go back to the slides. We generate our histogram of single cell level data. And here we're just using a microscope to scan along the device to do our cell counting and then to visualize levels of protein expression based on magnetic particle labeling. So then this allows us to provide accurate quantitation of protein expression at the single cell level, even in the presence of a billion-fold excess of non-target cells, and allows us to provide a highly accurate expression index for a protein of interest. And this type of quantitation is difficult to achieve with conventional fluorescence-based measurements, where you're using fluorescence to try to quantitate levels of protein expression. So on the left, you're looking at a comparison of two cell lines where the analysis is done by flow cytometry. You can see it looks great, but you need at least 1,000 cells to get data of that quality. If you try to isolate cells and then quantitate protein expression using fluorescence microscopy, you can see the difference between these two cell lines that express different levels of PD-L1 is really compressed and the resolution is lost. But now if we use cytofine, you can see we have much, much more resolution. And even with a very small number of cells, we can still resolve the large change in protein expression that you would be able to see with flow cytometry. So we've used this approach to look at a variety of markers in rare cells. We've done lots of work on surface proteins using the approach that I just described, but we've also worked out methods to look at levels of intracellular proteins, RNA sequences. For those markers, we need to fix and permeabilize the cells, but for surface markers, we can work with live cells, we can recover them after the analysis. We get high levels of recovery and also high levels of viability. So we've applied cytofine to a variety of problems relevant to therapeutic development, as well as new disease screening approaches. We've looked at the expression of markers like PD-L1 and ARV7 in rare circulating cells in the blood to provide real-time information on the presence of cells within a tumor that might respond or be resistant to targeted therapies. We've also shown that we can visualize rare cells in batches of CAR T cells, of therapeutic CAR T cells, and differentiated human pluripotent stem cells, which is very powerful to ensure the safety of these types of therapeutics from lot to lot. We've worked with the head of on the diagnostic side of things. We've worked with the head of our mesothelioma screening program in Toronto, to demonstrate for the first time that myeloid cells proposed to be present during the progression of mesothelioma can be visualized in the blood of patients. And these cells can be used as powerful biomarkers to use for mesothelioma screening. We've also put this capability to work to enable unique types of CRISPR screens where rare cells are recovered in vivo to reveal drivers of metastasis and other types of disease biology. So with this platform in hand and a suite of powerful applications related to the ability to visualize rare cells, we've launched this new company about a year ago, Cellular Analytics, that will be developing automated instrumentation to allow this type of capability to be widely available. The company is also developing its own assays for the manufacturing of cell-based therapies and is partnering with pharma companies on the development of assays that enable therapeutic response to be predicted in specific subpopulations of patients. Cellular Analytics has been funded so far with about $1 million worth of seed capital. It's now kicking off a Series A raise, and so if this story sounds interesting to you, please drop us a line. So to wrap up, I just want to thank my group. This is an incredibly talented group of really creative scientists and engineers. I thank them for all of their hard work, and I really thank you for your attention.
Thank you so much, Shana. It sounds like you're off for another successful venture. I appreciate that. Next, we've got Dr. Ali Tanazli. Dr. Tanazli joined Flexergy in October of 2019 and oversees all commercial operations, strategic planning, business development, sales, and strategic partnerships. Prior to his role at Flexergy, Dr. Tanazli led the corporate-wide global strategy for healthcare and life science for Hewlett-Packard. After receiving his PhD, Ali worked in corporate development at Applied Biosystems, which is now part of Thermo Fisher, and built a life science business at Sony. He also currently serves as an angel investor and board member at various startups ranging from cybersecurity to oncology. Ali will speak to us today about microfluidics and hypermobility for Health 4.0. Welcome, Dr. Tanazli.
Hello, good morning, good everyone. Good afternoon, everyone. Thanks, Lynne. Thanks, StarFish, for the kind invitation. I will be speaking today about tools for new medicine. And as Lynne mentioned, I was in my past life at ABI, Sony, HP, have seen a lot, and we'll give kind of an overview, but all the next steps, what we can actually do in terms of shaping the future. First, we all know the Gartner's hype cycle, and we all, if you look back into the past 20 years, there had been hopes up at the beginning that microfluidics will be super, super big, and then, well, people go through different phases of the hype cycle, right? Great innovation, end of 90s, and then 2001, everything, or let's say 2000 until the crash of the newco. There's a peak of inflated expectations, and then when you look at market reports, so it's always really entertaining to study old market reports because you see basically how numbers have been projected. Didn't really happen that way, but I will explain you a bit why, because I spent quite a bit of time on strategic perspectives and thoughts and health economics, but I think there are some interesting, how should I say, opportunities out there right now when we look at those new things happening in the past 10 years, I would say, mainly in IT. And I will do, like in the next 5 slides, some sort of a technology preamble, because what's important to state, I think, is microfluidics itself is a great horizontal technology, right? But then it depends what you do with it. And there was always the quest for the killer application. Now, it's standalone. It went somewhere, but I think there's still huge potential to be unlocked. What's interesting now, in the past 10 years, many things happened on the IT space, also on the manufacturing space, and what we call today as Industry 4.0, basically that's the emerging of cyber-physical systems, is super exciting because it embraces all levels of technology and society. And just to give you some examples, so first, Internet of All Things, or Internet of Things, about 10 years ago, we started with the first IoT devices that, for example, all home printers or office printers you have, they're all actually IoT devices, just no one knows about it. So we have already great infrastructures in place and see how can we make scalable IoT happen. And then you have, and this will be important part for point of care, actually, I will lay this out later. And then we have also aspects of AI and machine learning, which are important to manage large amounts of data, which is important to manage decentralized data. I will speak about that. And then we speak about, or like when we look at digital manufacturing, then we actually speak of a very interesting asset here. It's the asset of democratization of things, which had been centralized before. So basically, when you look at normal manufacturing in a factory, manufacturing happens only at a factory, but now you can also manufacture things in a decentralized fashion all over the place. There's also an interesting example of democratization. In healthcare, we will see different other examples, but there's just one very illustrative one. And then ultimately, all this needs to be interconnected in a secure fashion. So cybersecurity is getting more and more important, especially if you want to make point of care happen. You need also to think of cyber secure solutions. Otherwise, the use would be very limited or let's say threatened. And ultimately, how can we use these type of different assets or technology concepts to basically establish new perspectives, new tools for medicine? Now, just to look at this technology, this tremendous technology, let's say, shift which happened over the past 10 years. Back in 2010, we had worldwide only 300 million connected devices. Now, within 6 years, by 2016, we had already 30 billion connected devices. And now you can expect we will be close, I think, to 100 billion connected devices very soon. And that's very important because the connectivity which is happening here was actually one of the missing links in the past when you look at point of care devices. And point of care, to me, is almost synonymous to microfluidics because the biggest market opportunity or monetization opportunity for microfluidics is point of care. So I see to some extent equivalent. And here basically, this was the missing link for successful implication, or one of the missing links for successful implementation of microfluidics, aka point of care. And I will speak a bit about that. And this talk gives you really more like an overview, and we can always connect offline because I'm dealing with those topics every day. What's also interesting is many ideas which we need for point of care are actually very old. For example, artificial intelligence, which you need if you really want to do successful point of care, originated back in the 70s, basically, quite a long time ago, right? And basically those concepts are along. But what's interesting is when we combine all those different technology assets, then we see very interesting blending of reality in which we, on one hand, have from the physical world, the different inputs, the different sensor inputs. We look at, let's say, test results from IVD. We look at step counters. We look at heart rate monitors. All those things are now interconnected with the digital world, with the digitization step. And then it gives a beautiful feedback loop back into the physical world, in our case, into the healthcare world, and gives us also new tools to monitor success of treatment. Now, I know I'm mentioning many different topics. That's by intent, because I want to show you kind of that everything is really getting converging right now. and that's super exciting. So we basically have the opportunity to reinvent healthcare. And so as of today, we are truly in the age of technology convergence, because on one hand, we have now all the tools available from consumer electronics. I think all of what happened in the past 20 years in consumer electronics is a lot, and we see great opportunities to really translate this now effectively into healthcare. Microfluidics also is around since the early 1980s, actually. For example, when you look at the printer business that started in the 1980s, it's all based on microfluidics, basically. And then you have all the capabilities around big data analytics, machine learning, AI. Those are all different technologies which have been separated in the past 20, 30 years. But now it's time to merge all those technologies together. And the reason why, and we need to merge those different technologies together if you want to understand biological complexity. So when you look, for example, at, let's say, developmental biology, at the metamorphosis, right, of a caterpillar into a butterfly, still not much of this is understood. And when we look at the human health level, like what determines human health, what determines human sickness, we need to look ultimately into many layers of omics, right? So we have, I mean, most prominent examples are genomics, proteomics, metabolomics, but there's much more to that. That means ultimately we need to look at all these different layers, also to have a better diagnostics. Now, it's like when you look at, let's say, better diagnostics is embedded in the overall opportunity to basically disrupt healthcare. Because what we have in healthcare today are a couple of mega trends which are important to consider if you want to look at successful new tools for medicine. On one hand, you have aging population. That means you have basically less physicians per capita in terms of effective capita. Basically, you have a shortage of physicians, need more physicians. How can we make physicians better accessible, or how can we make healthcare services better accessible? Then we have the issue of drug-resistant superbugs. Now, during this crazy year of the COVID-19 pandemic, we didn't hear much about superbugs, but this problem still is around. And you can also look at a bigger topic, which is infectious diseases overall. And you may recall, prior to this pandemic, we all in the industry had been expecting the infection X, which actually happened this year, obviously. It turned out to be COVID-19, but it's just one part of the threat. There are other threats in terms of superbugs. So we basically need to manage healthcare better, need more preventive, and especially more decentralized testing and monitoring solutions. And also this COVID pandemic, I think, also accelerated the adoption of digital health. Now it's kind of more and more common to have a telemedicine appointment with your healthcare provider, which is, I think, great. Along with that, payment for healthcare is an important topic. Payment models are more and more transparent right now. There's increased transparency, which is great. Then you have also increased emphasis on wellness. When you sign up for healthcare insurance, you sometimes even get a smartwatch to basically support you in keeping track of your health. And then you have basically the technology convergence, which is a trend outside of healthcare, but has now the great opportunity to disrupt healthcare, which then ultimately leads into decentralized healthcare services. And you'll see a couple of primary technology implications, which are all part of it, which all represents basically the different subcomponents of Health 4.0, which is a subset of Industry 4.0. Now, when we look at digital health, there's actually, I will not speak in detail to this slide, but what's interesting is we have in the US about 190 million individuals affected by one or the other chronic disease, which also, as a side note, is one of the reasons why COVID-19 in the US is such a big issue, because of many, many underlying preconditions people have, unfortunately. But if you help and think of better chronic care management tools, then you can reach this 190 million people more effectively, treat them better, and ultimately avoid hospitalization or improve the treatment models. And that's just one example how digital health can help, basically through better access. And I will also speak shortly why this is related to microfluidics. So there are basically opportunities to prevent and to have basically faster, cheaper tools for healthcare, and then basically to have a beneficial impact on treatment of diseases. And What we need for this as a foundation is increasing connectivity. So it's a blessing that we get 5G networks now, which is also important for implementation of telemedicine successfully, and all the tools how to manage big data. Now, we see today healthcare cost and healthcare structure overall is a big mess. And COVID-19, on one hand, exposed the challenges we have in our healthcare systems worldwide, and shows also the different issues around accessibility, cost. Like we have, for example, COVID-19 tests. Sometimes you get a provider bill for about a few $1,000s, or it doesn't cost anything. It depends where you go. That means there's a huge issue with cost, huge issue with, let's say, transparency with the aging population. You also have a huge issue with insurance premiums and deductibles. So there are big problems, basically. Now, the upside is, as bad as COVID-19 is, from an overall public health standpoint, it's a wake-up call, because COVID-19 accelerates now the adoption of decentralized healthcare services. And one constituent of decentralized healthcare services is basically point-of-care testing. Now, what we want to have ultimately is in healthcare to switch from today's standardized reactive in-person care where you have to walk to, let's say, physician office. Like we want to move this to a personalized preventive and remote care. And that's, I think, very possible. And we have different tools for that already available. And I think emphasis on diagnostics is very important to address the healthcare issue. The US healthcare market is in total a $3 trillion market. Worldwide, we have an $11 trillion market for healthcare. And what's interesting is, and that's why point of care and microfluidics are ultimately very important, is that diagnostics amounts only to about 2% of the total healthcare spend. However, the medical value of the diagnostics, that means the impact of diagnostics, is more than 60%, because if you get a certain diagnostics, then it puts you on a certain track for hospitalization, for treatment, for even surgery and whatnot. That means diagnostics is very important. That means, on the other hand, you also know that lots of dollars are being wasted in healthcare overall. And so one aspect of this is, from a health economics standpoint, is when you do diagnostics more often in a better accessible fashion and in a more affordable fashion, then it will benefit the overall healthcare spend and lower the total healthcare expenses. That's the ultimate goal. And that's important, because when you look at today's medicine, what we have today is imprecision medicine. So basically, you have, for example, a drug which may work in a given percentage of population, but doesn't work in other people. And that's really a big issue, I think. This standard approach where you think one size fits all, or one drug dosage works for all people, it's wrong. Ultimately, you want to stratify this and really have a precise, personalized medicine so that everyone gets the ideal optimized treatment. Now, you have your different levels for IVD. If you want to do ubiquitous testing, it needs to be very low cost in order to be truly democratized. It needs to be at the point of need, for example, in retail clinics. It needs to be AI enhanced, because very often you will need to interpret different results, different clinical data at the same time. And then you can use this basically also for improved treatment ultimately. I will just speed up it a bit for sake of time. And when you look into Health 4.0, I think it's highly driven by hypermobility and microfluidics. Hypermobility is the capability to do edge computing, to do basically analytics, data interpretation on site. It means you don't necessarily need internet. On the other hand, you want to have connectivity to basically connect it with electronic health records and, let's say, clinic, LIMS systems. But I think what you really have now is an opportunity by decentralization of health testing, enabled by nano and micro technologies, to be able to focus on prevention and to personalize treatments. And here again, the key trends shaping healthcare are, on one hand, convergence of technologies. Customers and patients are more and more empowered and engaged more, and also technically, I think, more capable to think of testing. And especially, again, I think COVID-19 raised awareness big time. Regulatory changes are going on. Approval cycles are much faster now. And I think entirely the let's say, healthcare industry is more open to new solutions, and ultimately the IT, let's say digital, revolution from IT is an important contributor. Now, why do I do that? And so I've dealt with these topics for the past 10, 15 years, basically, since I joined corporate world. And what's interesting now is, it's like when I look at all these different, let's say, problem statements, the reason that I joined Fluxurgy is that Fluxurgy is developing a platform which is truly unique in terms of speaking to many of those things which is being addressed. For example, one reason of why point of care systems to date didn't have the full market penetration was due to incomplete panels. That means if you needed, and that means when you need it for a certain disease area, let's say an immunoassay and the cell counting at the same time, this wasn't going to happen at the point of care. It means you still needed to send it to the central lab. So we are working here on an analyzer platform which is capable of being multimodal, basically addressing cells to molecules, and also allowing a multiplex panel configuration so that you can really think of syndromic panels. And what's also important is to have it user-friendly, because ultimately you want to have a clear wafer for your instrument. So we are working right now on numerous applications in acute care as well as chronic care to basically address those needs. And then ultimately the goal is to reduce the overall healthcare cost by better accessible testing. And what's also important is, as soon as we have the data, especially for multimodal test results, you need to be able to interpret on-site to provide instant on-site results to the user. That's very important. On the other hand, you also want to communicate in a cyber-secure fashion to the, let's say, servers of hospitals, LIMS systems, or apps of retail clinics. So what we see ultimately is, and that's part of our vision at Fluxurgy, is we see the entire diagnostics as a continuum of different states of health. It's ranging from healthy through symptomatic diseases to chronic diseases. And what's interesting is each of those stages, from healthy to chronic sick, gives you the opportunity to monitor your health. Okay, very good. I think
Mike, we lost some slides.
Yeah, we lost the slides, but this was already my last slide, so basically it's a good timing. But here you see basically how everything is coming together. You see the stratification, you see the multi-omics, you see basically how you want to look at different stages of your health at different healthcare metrics, and basically enabled through AI engines to really interpret your results, and then have basically one analyzer which is multimodal, basically being able to address different sets of biomarkers and to enable personalized therapy. So thank you very much for your attention.
Thank you so much, Ali. Greatly appreciate that. Very, very interesting. Next, we have a revolutionary in the field of precision medicine. Kevin Rusovsky is an American biotech entrepreneur, engineer, and healthcare visionary who has dedicated his 30-year career to transforming medicine from reactive sick care to preventive and personalized healthcare. As good as the science has been, his playbook for business operations is just as transformative. His 2017 IPO of Quanterix, a company digitizing biomarker analysis to advance the science of precision health, was one of the most successful IPOs for a life science tools company in recent history. In today's discussion, Kevin will share how he sets up his framework for making great science into great business. And it's all about the culture and how you can do this too.
Thank you very much, Lynn. I really appreciate that introduction. And I think this should be an interesting opportunity to give you some insights. I'm noticing that my slides are not up though, so I'm probably doing something wrong.
We'll get, there we go, mic on.
Yeah, it's showing, my slide deck shows Dr. Tanzilali is up.
Yeah, that's still my deck. I think you may want to switch the file because my file is a separate PDF. You may want to go to the general StarFish webinar.
Mike might be able to help here get these slides up.
Okay, so now slides just popped up. There they come. Okay. So first of all, I would say, I'm really blown away by these presentations, and there's been so much insight and perspective. And I apologize if this is boring to this audience, because to me, the audience does have an incredible level of technical capability, and a lot of where I'm going to take this, even though I'm a mechanical engineer and I love the technology, is more towards the commercialization side. I've been involved with microfluidics for quite a while, maybe 15, 20 years. And I had a major collision with microfluidics when I took over as the CEO of Caliper. And I'd like to talk about a procedure and approach we use called DiFi for disrupting and mostly commercializing these technologies, and talk about COVID and the promise that we think these technologies and microfluidics holds for it, and also the whole new testing paradigm that's evolving. And as you heard, we did sell Caliper to PerkinElmer, and we went public. And then I'm on the board of 908 Devices, which is a handheld mass spectrometer, same founder as Caliper, and it's a great technology. And then Quanterix is somewhat another advance. And the lab on a chip over here on the right actually belongs over with Caliper, not Quanterix. But our valuation, we've put $400 million we've raised in the last 4 years into Quanterix, and making a lot of advances, and microfluidics will be key there as well. But basically, it starts with the opposite, meaning that you don't have a technology that you then try to find a way to move into the market. This starts with a greater purpose and seeing what the key headlines are you're trying to move, and then building an ecosystem. In this case, it's precision health ecosystem, powering precision health. And then recruiting the right culture of people and leadership, then building a strategic roadmap to try to move those headlines, and then figure out execution. So it's upside down. So I started this foundation long before I went into Quanterix, because we're trying to advance the field of medicine, where today we see United States having an incredibly high cost of operating in healthcare for the age and life expectancy that is being created. You can see that United States is kind of off the charts, and we're trying to work hard to move it up to the top by really increasing the accessibility and focusing on technologies that allow us to see disease long before symptoms. And that's exactly what Quanterix's technology, the same founder that founded Illumina, founded Quanterix, David Walt, and it's really a way to get the same sensitivity that you have with molecular testing, DNA and RNA, to proteins, which we think are more phenotypic. A lot of identical twins are born with the same genes, but yet longer term, their protein expression could be very different. And one might grow up to get cancer, while the other one grows up to get heart disease based on the way they live their lives. And so looking at very low invasive, high intel, early detection is a lot of what these visionary approaches of powering precision health are about. And this slide just illustrates on the y-axis the invasiveness of the test, and on the x-axis when you discover that someone has an issue in the pathology of their disease. And you can see that the concentration of the protein goes down when you reduce invasiveness. So if you're trying to get an answer from saliva, urine, or blood versus a biopsy or a cerebral spinal tap, you got a lot lower concentration where sensitivity is needed. Same is true. Normally, proteins go up in concentration as you get diseased and as that pathology progresses. And it's like the two most lethal areas is cancer and neurodegeneration that seems to be very detected very late after symptoms present. And by the time symptoms present, you may not have much time left in your life. And so we felt like if you can get low invasive answers in those areas of cancer and neuro initially, that's going to be the best impactful to the world. And you can see the number of publications now that have been evolving in that landscape. And you can see that over the next, just like Ali just presented, there's a lot of diseases that are environmentally triggered that are continuing to evolve and are going to create some pretty profound cost implications for the world. The whole area of Alzheimer's is one area particularly that we're watching very closely. There's no drugs, there's no treatments. Many of the neuro diseases, even CTE, where a lot of athletes are getting, it's very hard to diagnose and it's very hard to treat. So we're going to talk more about how we think these technologies can evolve, starting with the ecosystem. And so we're saying there's a biomarker revolution that we're trying to inspire and incite with this Powering Precision Health foundation or association. We've been running it now for about 5 or 6 years. There's a lot of peer-reviewed publications now. There's a lot of pioneers that are part of it. But a lot of what we do too is we try for third-party peer-reviewed publications to kind of prevent the Theranos effect of being able to do something that no one thinks is possible, but to truly do it. And you can see all the sponsorships we now have down at the bottom. And key to this, in our eyes, is having the investors attend with all those thought leaders as well as the policymakers. So we'll have all the top investors in the world attend, and many of the companies that end up sponsoring that are trying to illustrate their technologies and ability to profoundly affect precision health need investments, and they need to get funding. And so a lot of individuals end up teaming up with investors by coming to this to further fund their large ideas and further make them work. And so the two big tenets of Powering Precision Health is early detection for the purposes of getting treatments that are more effective and less toxic because they're lower dose. And we've now found that there's a significant increase in the probability if a drug company uses biomarkers in the way they stratify patient populations and the way they recruit cohorts, they can have a profound effect on the probability that they can get a drug approved. I just was on the phone with Scott Gottlieb, the former FDA commissioner, who put a lot of the guidance in for what we call in the neuro area, using biomarkers for precognitive impairment. Long before dementia, if you can recruit patients in that you think and can show clinically have Alzheimer's using biomarkers, they're going to approve drugs based on bringing the biomarkers down in the concentrations of the protein long before symptoms. And it used to be you'd have to reverse dementia to get a drug approved. And so some of the largest customers like Biogen and Lilly and others right now are utilizing these technologies and approaches to bring in folks with Alzheimer's long before dementia and or MS, multiple sclerosis, Parkinson's, many of the neuro diseases. And the same thing holds true for all the other disease categories. Cancers, you bring in early cohorts, you have a much better chance for the drug to work. Second stage, though, is using the same biomarkers to prevent the disease in the first place by the way you live your life and using biomarkers to guide that. You can see all the adoption that's occurred in the last 4 years across almost every pharma now is using these biomarkers. And that's why the valuation of the company has gone so rapidly north, is because of that very rapid growth. But more importantly, the payers. I was really intrigued with what Ali presented. There's times that you have biomarkers that can see disease early and a treatment already approved, like the MS. There's 16 approved drugs. You can now see MS much earlier using blood biomarkers than using MRI. And so deploy now and get some of the cost out and extend life for the payer group. But if there's a biomarker and no treatment, use your member group. In this case, United Health Group's got 70 million members. Use them to try to recruit cohorts in the area like Alzheimer's. You got the ability to see it early, no treatment. Let's try to get the treatment approved with those biomarkers. And then if you have a treatment, but you just can't diagnose early enough, like pancreatic cancer, surgically, if you can get it in stage 1, you would have a treatment. Well, let's now go find the biomarkers by running large million-person population studies over 10 years where you're looking at biomarkers quarterly and trying to figure out across these disease categories, what was the biomarker that could have predicted it? So that's the way we're teaming up with the payer group to really advance this whole concept of biomarkers. So in the area of prevention, there's liking, there's wanting, there's needing, and we know that the food industry has created a lot of addictions, and they even talk about, hey, you can't eat just one. Well, that's not so good for you when you can't eat just one. Those addictions have actually triggered a lot of biomarker insights into these areas where there's subsidies today for sugarcane and corn in the United States that's significantly lowered the price. And now we've got people eating sugars that's 160 pounds of sugar per person per year. And we also are utilizing pesticides, growth hormones, and antibiotics in that food chain that's really significantly heightened many of the dimensions of the overstimulating the kind of the insulin growth markers and hormones that are triggering disease like cancer, heart disease. We can see a lot of these issues. And so that's why the PPH ecosystem is saying, let's go fight that. And, you know, the next step here is culture and leadership. We really have to have the spirit of small, the power of scale, and have all-in movement. You want employees that's like on the edge of their seats, really going after this stuff. And we use a lot of movies to inspire parallels, to create a lot of excitement and drama around what we're doing for the world. And we try to use in our execution a lot of these techniques to see things much earlier and to go after the elephant in the room and to really provoke transparency into the issues that might get in the way of microfluidics ever becoming a good technology. And in the case of Theranos, we know that there wasn't that transparency and it actually triggered fraud. So for us, we spend a lot of time with disruption, making sure we have a lot of transparency. Because it's hard when you're growing at the pace we're growing 50% annually. There's comfort, but then growth creates a lot of challenge. And many times, like COVID, the biggest issues we have in the world represent opportunities to really showcase the technology. And in the interest of COVID, we'll show you in a moment potentially a video around it. But what we have here is a very fast-growing company because of all these biomarkers across all these different disease categories of cancer, Alzheimer's, neuro, as well as cardiac. And that growth is really further involving a lot of investor interest. And basically, we just have miniaturized the ELISA using microfluidics. And so it's the third incarnation that I've played a role in my tenure in microfluidics, but each of them become billion-dollar valuations only because of the commercialization aspect. And in the case of COVID, we've got big issues with policy right now, getting the economy back and getting people safely back to work. But at the same time, we've got huge false positives and negatives and asymptomatic spread of the disease that the diagnostics is not picking up. And so we've spent a lot of time in this area. I don't know if there's a way to show a video at this point. George Stephanopoulos, who we were interviewed a couple years ago from him around some of the microfluidics for seeing concussions in blood. So here we go.
I was taking the test fully expecting it was going to come back negative, but in fact it did come back positive. And I just want to bring in Dr. Jen to talk a little bit more about this. And Jen, we've talked about this offline. So many relatively asymptomatic people out there.
Exactly, George, and I'm glad you're feeling better and Ellie too. But we've talked about the fact that so far the CDC had estimated that 1 in 4 people who are in fact infected with COVID-19 show no symptoms at all. Recent data just out of Iceland was able to test a lot more people because they have a much smaller country and they
because of the echo. Why don't we come off of that video? Perfect. I'll just close up. Basically, 40 to 50%, if you can get my charts back, 40 to 50% of the carriers of the virus actually don't have symptoms. And that's the challenge, is that all the diagnostics today can only be seen when you have symptoms. And so we think seeing it in blood, saliva, and oral swabs using microfluidic, which we now have proven with data, is a bigger impact for the world. And so now the challenge is getting these tests approved through the FDA very rapidly. And the NIH is now funding us around many aspects of that scale out. And this slide here basically shows on the right that the false negative rate before symptoms is actually 80%, meaning that 80% of the time, if you're told you don't have the virus, you actually could have it, based on the false negative rates not being able to be measured with today's diagnostics. We've actually now got data showing that we can see many of these asymptomatics or pre-symptomatics with the disease and changing that whole false negative paradigm. And this is an example of some of that data you can see on the left-hand side. We are able to see it inside of that window where today's technologies cannot see it. And then on the right-hand side, we're taking that same false negative and saying for the 50% of current virus holders in the United States that don't have symptoms, what you have is as much as 80 to 90% false negatives. So it's rare that you ever can even measure it with today's technologies. And that's why advancing our approach is, we think, a good way to go. So in summary, I would say it's important, and each time you're going after something like this, you really want to start with a greater purpose of what that is going to really try to solve for the world, and then build an ecosystem of the investors as well as the top thought leaders to bring credible science to bear onto the problem, and then get the right culture, use people, and let those people get unleashed so that they can really tell you with a lot of transparency what the challenges are, and create a real passion and inspiration to go after the big ticket greater purpose, build the roadmap, and then execute with a lot of high impact speed leading. So I'll stop there. Thank you very much for your time.
Thank you so much, Kevin. Really appreciate those powerful insights. Our last speaker before the Q&A is Dr. Lorenzo Gutierrez, who has over 30 years of product development experience in semiconductor, consumer electronics, and medical devices. Lorenzo has developed new fabrication techniques, materials, processes, and methods in commercializing cartridges and instruments. He specializes in overall system design, hardware engineering, integration, and translation of all assay workflow to consumable cartridges. At StarFish, the microfluidic cartridge development team in our Toronto facility. I'll turn it over to you, Lorenzo.
Okay, thank you, Lynn. Thank you very much for the opportunity to talk with you about a topic on device integration today. So I'll be discussing about the integration of microfluidic to medical device development at a high level, and we'll discuss particularly on the area of device integration. Yeah, I got an opportunity to witness the microfluidic development for the past 20 years. Having worked in Asia, particularly in Japan, Singapore, China, Taiwan, Philippines, and other countries with close ties with companies in North America and Europe. So I also work with microchannels, paper-based microfluidic, DMF, semiconductor, particularly with thin film, PCB, printed electronics, and some instrument design, as mentioned by Lynne. So in this case, I would like to show you some kind of 20 years of experience. So on the first 10 years, I could say that during that time, research and development for microfluidic focused more on simplified application and tests, usually for blood tests, immunomolecular assays, with a target panel like HIV, malaria, TB, dengue, infectious and others. So on the second part, like another 10 years, so I divided 10 years, aside from the simplified application, we also have emerged complex tests like multiplexing, high throughput for cancer biomarkers, stroke, heart diseases, some sepsis, also molecular assays, and flu infectious diseases, and so on and so forth, plus a single cell sample preparation, or these work in combination. Yeah, in the last part of the decade, we got a COVID-19 come in that will change a little bit of the infrastructure and the ecosystem. But going back to 2003, I was in Guangdong, China during the time that SARS came in and infected the place, and most of my team there was infected with the virus, and the government told us that we have about 14 days to live, and I thought that would be another pandemic, but thanks God it did not happen. So the point is, at a given point of time, there's so many other infectious diseases that will come in to the time zone, but COVID-19 is the big one. So this one's going to another 10 years, about say 2020 to 2030. So I really don't know what will happen, but I guess COVID-19 make a good introduction for what happened. So I call it decentralization and personalized, and I call it like consumerized medical device, or Uberization of healthcare. This will involve like molecular and immunoassay that you could do at home, if it's possible, a new detection technique, and also integration of real-time data analysis and prediction. So there's a portion there that artificial intelligence will play a role, and new assay, new applications, and new materials. Yeah, in 2019, before COVID-19 came, so StarFish Medical did a market survey. We reached out to over 300 microfluidic companies in North America, Europe, and Asia, just to check the the heartbeat of the industry. So I would like to share you some of the 3 highlights in here, key insights. The first one is we have identified target and users and application, like reference lab, hospitals, physician's office, bedside patient clinic, home testing, and remote areas, which is very important in terms of you design a certain application. Pain points and technical issues, like understanding the testing and quality control, manufacturability of the developed cartridge, interconnect as a common problem, assay translation and device integration, turnaround time if you are in the publication, and of course, scaling up. Another key insight that we gathered is that the market segmentation and development stage, wherein some companies work on a point of care or life science or other application, and some others as they're on the concept stage or in development or in commercial application. So product development for me is a sort of a race. So a race that you could move into a finished product. So this is a typical product development cycle wherein you consider everything like concept development, proof of concept, building alpha, prototyping for beta, pre-production, and scale up. So I put some kind of terminologies in front wherein you can maybe identify some of the terminologies there that will affect you, or maybe a terminology that will make you awake at night, or maybe a heartbreak. For example, sensitivity or variability of the result of your test, or some kind of profitability or repeatability of your cartridge. So these areas are the things that like affect our product development in general. So assuming that if you are thinking of an application or target products, as I said, the sales panel, the goal really is to have it commercialized, like either ClearWave or FDA approved. So we'd like to give you some tips on, a little bit to bring the gap between your application to your commercial product. So number 1 will be check the complexity and maturity of assay to be translated to microfluidic format. So this will reduce the risk and improve translation performance, ensure the end product performance, like indicators such as sensitivity, specificity, variability will be checked. And you can review the instrument and control system and detection. This is called a system integration. It includes means of detection such as optical, electrochemical, or PCR, or some other things. Also consider manufacturability, cartridge repeatability, and cost. This includes also the most important thing, usability, selection of material, process, and ancillary equipment. And lastly is the business aspect. So even though you do everything, but it's not profitable or it's not affordable, that's a big issue. Another consideration is the intellectual property, freedom to operate, and a lot of regulatory matters. The next question is how to integrate microfluidic device to the process. So I make this diagram, say, if you are thinking of a project, say you are on the life science research or university, or you got a simple man that you got an idea, or a company spin-off, then the first step is to have a proof of concept and test completed. And the question is, did the concept work? If it doesn't work, then don't go forward. So if the concept works, then you can go into a series of steps for the device integration. The first one is product definition, wherein we can build the minimum viable product and develop the concept. In this stage, as you can see in the green color, we will review the assay workflow and the test result, as well as some other components that will influence the concept and also product definition. And also we'll do some kind of prototyping here. So the next step is translating the assay workflow to microfluidic and product testing. So we'll include now your fluidic handling and microfluidic, which are topic for today, and some optics and imaging and error detection as a part of the instrument and reader. And the next stage will be alpha prototyping and verification testing. We can also involve the manufacturing and the regulatory, doing some documentation and submission. The red line on the circle, or it's like a red line there, that is a system approach, meaning that even though you are on the step 2, you can still work with manufacturing because you want to see early on that your product will be manufacturable, or what are the regulatory requirements that you need so that you can solve it earlier. This will ensure that the device integration and scale-up will be successful. Okay, another thing is that microfluidic has a product subsystem. It includes the assay, which is, as we call, chemistry. Second is the cartridge. It's either plastic or some other materials, or instrument, the reader. That's one that reads the result of your analytes. So the best thing to approach this is to have a system approach. So this is another diagram that shows a detailed discussion on the device integration. So as you can see on the left side, there's a chemistry there, the cartridge, and the instrument, which is a part of the microfluidic system. And on the upper portion is phase 0, phase 1. It's a typical phase gate development process wherein there is a phase and there's a gate. And then this one is the feasibility wherein you would like to test whether your concept is working before going into the phase, another phase. And in vitro evaluation, if you got your test result is good, and going into clinical or preclinical or clinical trial. So in this case, there is a path going to the right, wherein the chemistry, the cartridge has own its specification and the interrelation with each other, and also the instrument. So this shows that there's an integrated approach that you can see with this diagram. So this is a very difficult, a little bit heavy diagram, but if you have some questions, this one, we can talk it separately. Okay, as a key take-home messages, I would like to give you some tips for successful microfluidic device integration. Number one, we should have a clear understanding on the intended final product. So as I mentioned, take a system approach and identify the critical parameters. Decide on the implementation strategy for appropriate materials, publication, and detection means. Second, test the real sample as early as possible. Explore reagent stability on the cartridge and adjust a strategy as needed. Third is align the commercial scale cartridge manufacturing process. Plan your supply chain of the system component to support clinical testing. And lastly, as much as possible, avoid shortcut, because eventually this will affect the quality of the result, increase costs, and worst case, you may sacrifice company's credibility. I guess that's the end of my presentation. Thank you very much.
If I can have everybody turn on their cameras and mics again, I would appreciate it. And thank you, Lorenzo, for that. We'll get to some Q&As. I'm going to pull one out. I'll start with one that showed up in the chat, and I'll direct these. It will probably be maybe to Shana and Kevin and Ali. Sorry, and also Steven. What are your recommendations for commercialization strategy for a microfluidic startup spinning out of a research lab? Should you go after investors right away, partner with big firms, or wait till clinical validation? Shana, you want to start?
Sure. I'm a big proponent of keeping things inside of academic labs as long as you can to really do as much of the validation as you can before you have the pressure of, let's say, venture investors or a strategic partner. And so that's what we try to do. We really try to de-risk in the academic lab. As it was just said, run clinical samples, really understand where the toughest parts of commercialization may lay. So that's what we do. It's hard to make that step from academic research grade platform to something that goes into a company, but the better the data, the better your chances are going to be.
Excellent. Steven, anything different to add to that?
No, I completely agree. I mean, waiting until you have the preclinical data, you're probably not going to survive that valley of death. I agree with the taking it out of the academic as fully developed as possible, because you're going to be in a new world of trying to commercialize that. The rule of thumb used to be 7 years from the publication to the device. So keep that in mind. You might shrink it to 5, maybe to 3. It's not going to be 6 months or a year.
Yeah, excellent. There was a couple of directed questions that came into the, that I think this one is going to use, Seven. What it was put up during your talk. What surface treatments were used in the cartridge, naturally or non-hydrophobic?
Depending on which cartridge, for the rapid hit cartridges, there weren't modifications, surface modifications. On the mega base of doing separations in capillaries, you have to put coatings on that or have a gel that doesn't have electroosmotic flow. So it really depends on which cartridge. I try to avoid having to change the surface in a microfluidic device if possible. It just leads to very complex manufacturing at times.
Another one that could probably, any of you could answer, why are air bubbles a problem?
They are a problem because they will change your flow and they can block a channel. They can interfere with mixing. You can also use them to mix. So again, they can be friend or foe, but typically they're foe.
Okay. Another one that came through was, is there special needs on electronics devices controlling or sensing the valves and other related devices? I think this was still during your presentation, Seven.
In the ones that I showed, we didn't sense the valves. If you're going to have 120 valves and 120 sensors, you got a really complex system. So we spent 2 years going from our glass devices to the plastic ones because things have to work each and every time. You know, your failure rate has to be that 99.999 or greater. So having sensors, then you have failures of sensors. So you're much better to develop something that's reliable and then test the heck out of it.
Okay, thank you. How is cytofine different from current methods to find cfDNA and liquid blood samples for oncology diagnostics?
So cytofine actually looks at intact cells. And this is a big difference from looking at, let's say, fragments of DNA in the bloodstream. You know, I wholeheartedly agree with the comments that Kevin made about the importance of looking at protein signatures and looking at phenotype, not necessarily genotype. And I think for liquid biopsy and therapeutic decision making, you want to know what's going on with the tumor at a functional level, not necessarily just a genotypic level. So that's what we're doing that's different. We're reading out functional information about cells in real time using measurements of protein expression.
I'm seeing that, thank you very much, Shana. I'm seeing we're not going to get to all the questions, but I just want to assure the people that have asked them that we'll get the speakers to answer the questions after, and we'll send out responses to you. A question that has come through for Kevin is, if there has been any correlation established between the input biology, i.e. food sources, and the effects on biomarkers or protein expressions in humans. In other sense, could the prediction move further up?
Yeah, absolutely. I think we measure a lot of cytokines, which are the inflammation pathways. And so we see a lot of correlation for different types of cytokine fatigue and issues around cytokine. Certainly disease and drug side effects can be triggers of environmental factors that we see a lot of side effects from drugs. I think there's actually data that would suggest that side effects to drugs can be as much as the fourth leading cause of death in the United States, particularly when you start thinking of chemo and some of the lethal effects. But one of the areas that we've seen a lot of biology, because a lot of the work's been done in neurology and neuro, is concussions. And so there's a lot of evidence of neuronal death that sometimes doesn't recover. And we measure NFL, neurofilament light, is the kind of like the engine light that says your brain is suffering neurodegeneration. Sometimes that's from a disease like Alzheimer's or Parkinson's, MS, ALS. Sometimes it's from events like the header in soccer. There's a lot of studies that have been done for football. A lot of studies have been done with cheerleading, just head trauma in general. And the levels of NFL, and sometimes those sustained levels, a lot of football, Division I athletes, football-wise, there's been a lot of NIH studies. Soldiers experiencing post-traumatic stress disorder through the blast, concussions from blast victims. So those would be the areas that the correlations have been greatest so far, but clearly in the area of cancer, there's a lot of opportunity looking at inflammation and triggers of inflammation from your environmental factors.
I just want to take a minute to thank all the speakers again. I really appreciate the really interesting discussions, and we will get these questions sent to you and get responses back that we've got in the chat. I'm sorry we didn't have enough time. And for those of you that are able, please join our September 18th webinar. It will have the creator of Shipamed, who's going to be speaking and getting a one-on-one interview with Scott Phillips, our president and CEO. So thanks again to all the speakers. Appreciate the participation, and I hope everybody has a great day.